right, thanks for waiting, everyone. I guess we can make a start. So good afternoon, evening, morning, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format, with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Typically, these sessions will run for about 50 minutes in total, and we will run this series throughout the year, generally on the last Thursday of each month, unless they overlap with the holidays. And hopefully you've just seen the new summer season shown on your screens. If you are interested in presenting a new method or software you've been developing or implementing, then please feel free to reach out. We are always happy to hear from the community. So before moving on, a few quick introductions. My name is Bethany Allen, and with me today I have Erin Dillon, Lewis Jones, Harry Drage, and Will Gertie. The five of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiological community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. If you want to know more about the Palaeoverse, please check out our website and GitHub organization, and hopefully you'll see some links in the chat to those. That's all from me for now. Erin will give you some helpful tips and instructions for this webinar, and Lewis will introduce this month's speaker. Thank you very much, and please enjoy the lecture. Over to you, Erin.
Awesome. So as Bethany mentioned, the format of this seminar will consist of a talk from our speaker, followed by about 10 to 15 minutes at the end that will be dedicated to Q&A and discussion. My fellow hosts and I will help field your questions and moderate this discussion. And please note that the webinar will be recorded, and it will be made available on the Palaeoverse YouTube channel and on our website. However, we will stop the recording before the Q&A period in case you would prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. You'll see a Q&A button. So you can use this to submit questions to our speakers. And during the Q&A session, we'll either read out your question or invite you to turn on your microphone in case you'd like to ask the speaker directly. There is a raise hand button. So alternatively, you can raise your hand and we'll call on you and invite you to turn on your microphone so you can ask your question. There's a chat feature. So currently you can only chat with the hosts in case of technical difficulties, but we'll allow you to chat with the other attendees during the Q&A period. And then there's a closed captions button that you can turn on to view the transcript at any time. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment, inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to the Palaeoverse Code of Conduct, which can be found on the website, and I believe a link is also being put in the chat now. So with that, I will pass it over to Lewis to introduce today's speaker. Over to you.
Okay, so thanks Bethany and Erin for getting us all up to speed on how this lecture series is going to work. Today, I've got the really great honor of introducing our speaker, Dr. Katie Collins, curator of the benthic mollusk at the Natural History Museum in London, the UK. So Katie is a paleobiologist specializing in systematics, evolution, and morphological diversity of the class Bivalvia, and is, I would say, the world-leading expert in the Cenozoic molluscan faunas of New Zealand. It's true, come on, Katie. Katie is deeply interested in shape analysis methods, including the geometry and measurement of spiral forms, which we'll be hearing about today. In their work, Katie combines 2D and 3D imaging and characterization, as well as descriptive analysis techniques to address key questions such as how functional diversity in mollusks has changed through time. So without further ado, we're really excited to hear from Katie today with a brilliantly titled talk, both round and upward, spiral, morphometry, and how to compare things that are the same but different. Please join me in welcoming Dr. Collins to our virtual stage. Over to you, Katie.
Thanks, Lewis. Thanks, everybody. Let me just quickly share my screen. Okay. Come on. There we go. Okay, hopefully you can all see my slides. So, why do we measure things? The That's kind of a fundamental question, I think, although I think a lot of us, we just skip to how can we measure things. But we measure things, I guess, because mostly of a desire to know something in a way that is standardized. And sometimes that question is very simple. Is how big is it? There are a bunch of different ways you can know how big something is, but that's a reasonably simple thing to measure, depending. But shape, which is kind of what I'm here mostly to talk about, is a bit more complex. So you can take dimensions and you can get aspect ratios, but the aspect ratio of an elephant is not super informative about the elephant. But certainly that's a shape. You can get a shape out of an elephant that way. You can take measures of more specific bits of anatomy. You could take trunk length of an elephant and trunk length of a tapir, and you could compare those. Or you could do a landmark analysis. You can put points on the elephant, and then you can compare it to another creature, perhaps a hyrax, and you might find things that they have different and in common that you wouldn't get out of measuring how big is it, or what is the aspect ratio of an elephant versus a hyrax. So these are all measures of shape, and they're all valid to a certain degree, but they answer very different questions. And when thinking about morphometrics and morphometric methods, I guess I always kind of come back to something that one of my geology professors always used to say, which is, when the only tool you have is a hammer, every problem looks like a nail. And I think sometimes when we learn a method for measuring something, that's the one we're going to use, kind of almost to the exclusion of any other, regardless of what question we're asking. So my foundational excuse for this talk, because as Lewis said, I focus mostly on bivalves. I primarily work on bivalves, which are very tractable to 2D geometric morphometrics, at least below the family level. But I got very interested in this particular group of snails, and particularly this very hypervariable species, Palaeoverse vermis, which had been separated into about a dozen species, named species, and considered perhaps as a biostratigraphic marker before my very sadly missed colleague Alan Bew pointed out that this is a direct developing species, and these forms all integrate to each other. And so it's some kind of like environmentally mediated plasticity. And I wanted to know, oh, cool. I was like, right, great. This isn't 2012, so a while ago now. Cool, let's take these forms and we'll look at what was going on in the environment at the time, and maybe we can find out a link and sort of go, okay, what's controlling form? And unfortunately, I still haven't answered any of those questions because I got quite stuck on this question of spirals, and I'm still there. But crucially, I could have performed an outline analysis. I could have performed a landmark analysis. I would have got out something that looked like a result. But to do it, I would have had to make compromises, and I don't think I would have trusted the results. I'm about to show you why I would have had to make compromises to do this. So if you do geometric morphometrics, you can zone out here. I'll alert you when we come back to something that's a bit newer and a bit more interesting. But just in case anybody here is not a morphometrician or has forgotten their Zelditch at all, geometric morphometrics is about placing landmarks on points. And it works really well within species or genera or wider, depending on what kind of animal you're looking at. But looking at snails, it doesn't work super well above the genus level. And because of the way that we photograph snails, generally it will also return you results that reflect taxonomic identity. So just to take an aside here, right, like this sagittal section of the snail, like that is not a profile that the snail presents to its environment at any point. It does not live stuck up on its apertural end and just going along like that, right? Like this plane that we see, this sagittal section, is effectively what it presents to the seafloor. And this has nothing to do with mediating how it interacts with its environment, except in some very small ways. And the landmarks that I've been able to place on there, they look like good landmarks, nice Bookstein type 1 junctions of things, until you realize that those diagonal lines that go up the spire of the snail are just what you can see of one continuous spiral. And so in fact, these sutural landmarks are just very widely spaced semi-landmarks along a very long curve. And the other point about this is that the spiral itself causes complications. Snails in general, in a population, will often, but very much not always, have the same number of whorls per individual, per an adult, right? The second they start varying how many whorls they have within a species, you can no longer do this kind of analysis on that species. You have to find a different way to do it. You have to drop some landmarks somewhere, maybe place only one at the top and then one on the body whorl. Like, you'll have to find a compromise. And the wider the taxonomic catchment, the more morphological resolution you start to lose. And that may work for you for the question that you're asking, but it may not. And so to take this to almost an absurd extent, here are a bunch of snails I placed these sutural landmarks on, and you can see that they fail at that first hurdle of GM, which is, do you have the same number of landmarks? No, can't compare those. If you then decide you want to do an outline analysis, and there are several papers that have done this and have effectively put a snail on a flatbed scanner and done an outline of it, again, this is not a particularly useful outline for a snail. This doesn't tell you anything ecologically. That's not the way at which they attack the planet, as it were. And so you are straight up just missing important bits of morphology. And this is wildly unsatisfying if you're trying to answer questions about either development, history, growth, or function, environmental effects, anything like that. These kinds of data will not help you answer those questions, even though you can undoubtedly set up an analysis that looks like it's doing it. So the title of this talk talks about how to compare things that are the same but different. And this is what I'm getting at, right? Geometric morphometrics wants landmarks or curves that can be landmarked. And they need to be explicit homology. So they need to not only be the same structure, but be a structure you can put a finger on, or, a bit smaller than that, put a stylus on, put a pin on. You need to be able to say that point, exactly that point, and only that point is homologous to the point that it's equivalent to in the next specimen. Exactly that point. Or the curve. You can do the same thing with a curve, and then those are semi-landmarks and you lose a degree of freedom. But you're still describing a very discrete structure. There are homology-free methods. There are methods that deal with surfaces. But what do you do when you have a homology and it's not like the whole thing, but it's not specific, and it's just that weird, wibbly gray area of homology, not homology? How do I measure it? And I think, unfortunately for me, that's exactly where I live because I work on mollusks. They are a great example of this. The molluscan shell basically is homologous between classes, but even within classes, not a lot of landmarkable structures are shared usually to be useful in any kind of broad state. So for instance, two examples here, you've got a nautilus and a nipponites, which is an ammonite. I don't know if you can see my mouse, but here's the nipponites and here's the nautilus. And I wonder how we might landmark those such that we get a good answer out of what we end up with. The other one a little bit closer to home for me is bivalves. This is Dendostria frons, which likes to live on gorgonians. And here is, in the middle is carnivorous cuspidariid cardiomya. Again, homologous structures, very difficult to compare. Just because you're homologous doesn't mean you're landmarkable. And to take this really to extremes, these are all muricid gastropods. So these are all in the same family. They live in many cases in very similar ways. They do a lot of similar things. And I really would defy you to come up with a nice detailed set of landmarks that captures their form without being distracted by some of these extraneous structures, but also maintaining the morphology that is there, the homology, sorry, that's still there. I'm going to stop right now, though, and say I'm not suggesting we throw out GM. Don't panic. It does work, and it usually is the tool you want. It's a very good tool. But if you read morphometric literature and you're interested in mollusks, sorry, it's been a long day, you might find it strange that you start stopping seeing any kind of wider analysis of snails, particularly, or mollusks at all, above the genus level. And certainly you don't see analyses that cross classes in mollusca, which is weird. They're a really good group of animals for sort of thinking about, like, how do we look at tempo and mode of evolution through the fossil record? Well, they're in damn near every sedimentary rock that has any fossils in it. They fossilize beautifully. Their shell is very informative in terms of what they're doing for a living. They've been there 500 million years. They're on all of the major land masses. Like, really, why are we not exploiting this data set? And what it boils down to is the damn things are hard to measure. So when I first wrote a paper about this problem, I called it Going Around the Twist, which is basically how I felt at the time after having spent about 10 years staring at this problem. But what I'm going to do today is run you through that paper and the analyses from that paper, and specifically how to use the Markdown file and tools that came with it in case you want to run this analysis of your own on your own. But also want to present an update because in the last 2 years, Roma and I, so the first two authors of that paper, have been working on a moving reference frame model for attacking this problem, which will allow us to. So this one works on snails. This one, going around the twist, works on snails, and it specifically works on snails with a coiling axis that you can identify. The new method, which I hope we will call both round and upward, because I think it's a much more cheerful title, will be able to incorporate in the same space gastropods, bivalves, scaphopods, and cephalopods. And when I say cephalopods, I do include Nipponites. So we will see some of that later. So coiling is a fundamental property of conchiferan mollusks, cephs, bivalves, scaphs, gastropods. The shell is what's homologous between them. And if you go Googling idly for mollusk morphology, you will eventually, won't take you that long to stumble across this paper, Route 1966, and this diagram, which to me is the most beautiful thing in all of paleontology. I love this diagram to distraction. I think it's honestly the most wonderful thing. And it treats coiling as a descriptor of form, but it is a theoretical space. This was not built from real data, and we'll get into that in a second. But I said naively, in 2012, cool, no problem, we'll just go take those measurements. I don't know if any of you have ever tried to take those measurements, but it gets interesting real fast. So measuring spirals is hard. And I've been leading you towards the answer to the first question here being no, it is not particularly meaningful to measure a coiled shell using a length, width, and a height. Ideally, to understand the coil, you want to measure the coil itself, because you can have two spirals of very different spiral parameters that occupy the same bounding box, right? But our understanding of how to do this, the analyses that Raup founded his work on, were in turn founded on a set of analyses that were done around World War I. And measuring spirals is very difficult. And so fairly logically, what Moseley and then Darcy Thompson did was to go, okay, we'll measure what we can and then we'll extrapolate, because we understand how spirals work, because mathematicians exist. We can go back to our Euclid, we can work on the geometry of this. We know this, we got this, we'll just take the measurements we can and then we'll extrapolate. And that's pretty reasonable, but that's why the Raup space in turn is a theoretical space, not an empirical one. They built the model first and built the space on that. The only parameter that's particularly easy to measure on spirals is out. So on a snail, if you know your snails, you're talking about effectively the shoulder or the whorl width for each of the whorls. So snails step out like that. And it's easier to measure on some snails than on others. So this is where the trap starts to be set for you. It hasn't quite been sprung yet, but this is the beginnings of the creaking of the bear trap of this method, which is it's very easy to measure these profiles on a Turritella, reasonable on a Semicassis. Bucicon goes the wrong way. Does that matter? We're not quite sure. We don't know, but we'll keep going. We'll get our calipers out. Distorsio likes to grow like a figure of eight on its side. Like each one of those whorls laps up in a curve on another one. So you start to question, where should I be taking these measurements? This is a little bit tricky. And then you go to Murex and you suddenly realize that the exterior of the shell is not helping you. It's getting in your way. And then eventually you get to a cowrie, at which point I imagine you probably throw your calipers at the wall and storm off in disgust. But just enough snails got measured to make this generalization that underpins the Raup cube and underpins most theoretical shell morphology. Not all of it, but a significant proportion of it, which is that snails are generally or always logarithmic helicospirals. This is the big assumption. This is the thing that underpins the cube, that means that the reason that we don't use the cube as a morphospace because things don't go in it is because of this. The logarithmic helicospiral, so the form of the shell, comes about if you have isometric growth and an exponential growth rate. And this is true of the outward parameter, so as measured from the outside of the shell, which we'll get into in a minute, but we don't have any proof that it's true of the other parameters. What other parameters, you say? Well, snails aren't lines, they are tubes, they have a thickness, they have an inside, they have an outside. And so Raup, as well as having outward, had and downward, so W and T, had two more parameters, which are the distance of that tube from the coiling axis and the shape of the tube, although in practice he mostly modeled that as a circle so that he could just give it a radius. So I love this quote. The Raup cube is the very definition of a useful model, but what is wrong with it? Well, Thomson and Raup both of them state this explicitly, that the mollusk shell, the snail shell, is self-similar. It's the same shape all the way down, and it grows the same way all the way down. And if you have looked at a snail for more than 30 seconds, you probably know that that's not true. It is effectively the molluscan version of the infinite plane of uniform density, right? We know it's not true, but it's convenient. We'll just run with it for now. We'll get ourselves started, and then surely we can come up with some terms to deal with that later, except we haven't. But so here's three illustrations of exactly why self-similarity is not true. On the left-hand side, you've got a beautiful volute, which shows a very nice disjunction between the coiling axis of the protoconch, the larval shell, and the coeloconch, the adult shell. And then a very nice figure showing growth in strombus, showing that the shell does really change shape as they get bigger. And then one from my 2021 paper where we looked at the relative position of the shoulder of the whorl, the size and shape of the aperture within that whorl, and then the centroid of that aperture. And we show that they shift relative to each other through ontogeny. So taking exterior measurements of whorl profile points is not necessarily getting you the same data at each one down, which is kind of a problem. That's why self-similarity seemed to be right, because they were taking these measurements from something that was actually changing under the hood. The other thing is this exponential growth rate. So you can eyeball the self-similarity yourself. You kind of know that that's not really going the way it's going. But in order to check the assertion that all four aspects of growth that need modeling are growing exponentially needs measurement to check. And unfortunately, you do have to get at the interior of the shell to do this, either using a rock saw, which we did, which is difficult if you need to negotiate with a museum curator, because speaking as a museum curator, generally, if you tell me you're going to put my specimens through a rock saw, I say no. And sometimes I lock the door. Or you can use a CT, which is great fun and very useful, and the specimen survives and everything is wonderful about it, but you do have to have a CT or access to a CT or money to access a CT. And of course, not everybody has those. Unfortunate. So we did both. We used some specimens we cut up with a rock saw, some specimens we used CT for, and we used landmarks, our old friends' landmarks, to capture the apertures at 180 degrees around the spiral each time so that we could get all of the Raupian parameters that we wanted. And then could test them, which is what the paper is about. So we have a fixed reference frame model, and we modeled the rate of change of each of these four parameters, W, T, S, and D, along theta. So that's the distance around the spiral. We tested them against linear log X power law quadratic models, and we picked a best fit for each rate parameter. And now I'm going to change what I'm doing. So I'm going to end the slideshow and I'm going to open my browser, and hopefully you should now all see my processing workflow. Hopefully someone get on the microphone and tell me if that's not what you're seeing, but hopefully you can see my browser and this processing workflow. So this paper, what we gave for people to use, because it wasn't super complex and we hadn't really developed anything that was de novo, brand new, and needed a function or a package. We just pulled together a lot of things that existed. So we went, okay, we're going to do this as a markdown. So in order to run this file, you need a TPS dig file. So this is TPS dig. You may or may not know it. So you take your section, your images of snails, and you bring them in here. And you place landmarks. So there's a landmark there at the top of the coiling axis, a landmark at the bottom. And each of these apertures is digitized with 50 points, equispaced. TPS dig will equispace points for you, so you don't have to worry about that. And yeah, so there's about 180 of these things in here. So this is what we did with that. So you take the file, this file, and you take the images that underpin it. And you also need to have run a shape analysis on all of those apertures. So we did a Hangul Fourier shape analysis. And so the data file for that looks like this. Each row is one aperture of one specimen. So there's about 10 apertures for this one, 128. So that's one specimen that I've just highlighted there, apertures 1 through 10. And these are the harmonics. This is the output of Hankel-Fourier, but you could also do it as a landmark, a semi-landmark analysis and run a GPA. So what you would put in here in these columns is the output of your GPA. So there will be the aligned coordinates. That's all you need for it. Or the PCA, I think it's actually PCA scores, but the markdown tells you. So we then run it through. So if you put all those things in the right folder and hit knit on this, it will run it for you, which is nice and easy. Unfortunately, it's not thrilling for me to walk you through the method because I just ran the data set, but it's fairly well commented. There's a parser for the TPS. So if you use TPS files, you know there's a parsing function in Geomorph. This one is a little bit more complex because it also pulls out the keywords from the specimen info dialog box that you enter. It pulls out the scale, and it does a bunch of things that allow us to then build the metadata set using that. So it finds you the variables, it reads the number of apertures, and it converts the landmarks to millimeters rather than to being pixels, so that it's all scalable together. And then you extract the spiral by finding the centroids of each aperture and coiling it round. And I'm going through this trying to find you a nice plot because we did digitization error checks, of course. So you can, we did two of those with the rock saw specimen. We did 9 separate images of the same specimen. So I remounted and photographed 9 times and then did the digitization. With the digital things, with the CT scans, I took the same CT scan and I sliced it 10 different times to check that how much error is associated in just finding the coiling axis. And if you plot that, so this is the digitization error, is these two Palaeoverse vermis, so that's R, which is out, and Y, which is down. And then this is Tyler Spiral Coronata, which this is 10 completely new sections. So this is the error associated with just the sectioning, which is still pretty tight. So we were pleased with that. So then we modeled the shell growth, and you then have to make some input to this, right? Because we ran a bunch of different models. We ran exponential, linear, log, polynomial, and power, and you then have to pick which one you think is the best fit. So we give you 3 options. You get a residual plot. So you want a flat residual plot. So the power law is looking fairly nice for flat residuals. We give you R² values. Power law, again, is looking very nice. An R² of 1. How much more could you want? And we tabulate that just to make everything very easy. And then plotting relative likelihoods. Again, the power law is your parameter of choice. That's your model of choice. In order to test that it wasn't just like the number of apertures or how much power the number of apertures have on whether or not you recover the same numbers, we cut down the data set to just do four apertures per snail. And if we scroll down past all the code, we're still looking fairly flat on the power law. The R² is still strong, and the relative likelihoods are still strong. So even with a much cut down data set, the power law looks good. And I'm speeding up a little bit because I feel that I'm going to run out of time. For W, the outward parameter, the exponential turns out to be the right one, which is good because that's what our friends Mosley and Darcy Thompson and Raup found in their own data. We are recovering the same result. The outward parameter really does have that exponential rate insofar as we can tell. So that's nice. And the 4-parameter model shows the same thing, which is lovely. The D parameter, if you are familiar with snails, you know that this one is always going to be the dodgy one, right? Because snails vary wildly between having apertures that basically sit over the coiling axis. So the center of the spiral is like a snake, or they diverge wildly and have a huge umbilicus underneath them and they don't touch the coiling axis at all. So the idea that there's one single best fit model for all snails for D seems very unlikely, and that is borne out in our analyses here. Not one of these has a mean R² of over 0.5, which is not great. And the yes, linear kind of comes out strongest on the relative likelihoods, but that's not a great plot. So D, if I was measuring these parameters and I was trying to run an analysis and I was trying to see what might covary with aspects of mollusk shape, I probably wouldn't put a lot of weight on D. And then S is the one that's slightly different. We use our shape analysis and we run it through a PCA and take PC1 as a single, like a linear descriptor of gross shape. The nice thing about doing it with a PCA is you can then plot your PCA, interpret your PC components, and pick the aspect of shape you think is most important for differentiating taxa. So you can be very explicit about which shape parameter you are using, and you can test all your different PCs and you can see what falls out, and that's good fun. But effectively, what it boils down to is, and there's different ways of checking. We can check the model fit here. So the colored dot is the predicted and the black dot is what really happened. So you can see some of these are a terrible fit and some of these are very good. But what it boils down to is we now have effectively the 6 faces of a route cube, which is fun. And these are all snails. Okay, so again, because I feel like I'm running out of time. I will keep going. I will stop with this one and we will go back to the PowerPoint. So, okay, so that's running the going around the twist markdown. If you are dealing with a data set of anything that coils around the coiling axis, it doesn't have to be a snail. This would work on ammonites also if you have planar spiral ones or helicospiral heteromorphs. It won't work on irregular heteromorphs, but helicospiral ones, it will work. Or if you have, other things spiral, shark poos spiral, if you're into sharks, that could be an interesting analysis. I'm in conversation with someone who works on antelope. We're looking at doing antelope horns, anything with a spiral. But lots of mollusks don't fit around a spiral. They just have other things. They're either a simple curve, like a bivalve or a scaphopod is usually just like a little part of an arc. Or maybe you have something that definitely wiggles and squiggles, but is not a regular coil at all. If you dispense with the coiling axis and just deal with the curve of centroids through the tube, then you can get somewhere with being able to compare those in a central kind of a way. And the way we've thought of doing this is using to characterize the curves as tangents and then to think about their yaw, pitch, and roll. Mostly in practice, yaw and pitch. So here are some simulated examples. So my colleague Roma basically did, in the finest tradition of theoretical morphology, he generated some shapes that he thought kind of looked like snails, and then said, what do they look like when I plot them? So here's a spiral that's roughly analogous to any sort of bog standard snail, and you can see that pitch holds fairly steady and yaw goes up and down. Makes sense. This one he generated because he thought it looked a little bit like Nipponites, which is rather our white whale. And you can see that not only does yaw have some stuff going on, pitch is going up and down like the Assyrian Empire, to quote Monty Python. Roll, as you can see, stays the same the whole way. This is because we're doing a single line. If we wanted to look at yaw explicitly, we would have to characterize the tube with multiple lines of points or sections of points, and you'd have to have those points be effectively semi-landmarks along the tube, so the same number of points. So to get roll to be informative, you'd need to be able to trace like the same point along the tube all the way around, which is kind of analogous to the way Savatsi was working on moving reference frames in the 90s, which is very cool. If you haven't read those papers, I highly recommend them. So you need 3D data capture for this, which is definitely the hardest part. Extracting centroids from strange objects that are not all very similar to each other means that we weren't able to find one single method of doing it. You can either use the same, if you have a coiling axis, you can repurpose the data that we used for the previous paper. Or if you're using something like a bivalve, what we do with that is we've been landmarking growth lines in 3D and then taking those rings of landmarks, one per growth line, finding their centroids and using that to plot the curve of centroids through. Because as you know, bivalves generally just go, they're just like a little cap or a soup bowl or some other kind of a shallow shape. But if you look at them, they definitely do spiral. It's just they never quite make it all the way around the horn, or they usually don't. Some of them do. The third way, which is kind of the new way, which is why I was swearing at my computer earlier today, is if your tube is long enough, so if you have a snail with a couple of way it whirls around, or you have an ammonite, something like that, you can use a technique from CompSci called mean curvature skeletonization, which will speed you up massively. So this is, data capture 1 using 2D and landmarking, data capture 2 using 3D landmarking and growth lines. And then number 3 is this being able to find the centroid of the landmarks in Nipponites. And I'm going to pop out once more because I want to show off this Nipponites just for a second. So this is a CT scan of lovely, lovely Nipponites mirabilis, which is a wonderful name for it because isn't it a little miracle? I spent some time cleaning this out. So unlike most ammonite folks, I was not very interested in the septa. I ruthlessly cleaned them out of the scan, and then I was able to do this. And I don't know how well you can see it, but hopefully you can see those are all the centroids from that shape. And so now those are ready to go back in with the rest of the data set, which is fun. So this is about where we've got to. I'm going to now show you where we are up to with both around and upward. It is very much a work in progress, but I'm feeling pretty positive about it. So this is another markdown. It will probably be a markdown when we publish it, again, because we haven't written a bunch of new functions. There isn't a bunch of package-worthy code to make this into. It's a workflow. So we're presenting it as a markdown. I should say I've suppressed most of the plots in this because it was plotting every single thing individually and it was taking an amazing thing. But what this does is it pulls in the polar coordinates from things that you've got polar coordinates on. And it then, those describe a 2D plane, right? Because we digitized them at 180 degrees, 1, 2, 3, 4, down using a 2D image. In order to get those to be a whole bunch of centroids around a curve, around a spiral, instead of a zigzag, we then have to move into a polar coordinate space, use a spline, and then transform back into a Cartesian space. So it's good fun. It took us a while to work that out. It took us a while. I took some time, and then Roman was like, oh yeah, I can do that. And then 5 minutes later, we had code because he's a wizard. The other thing we can do is to take in the specimens that we did, like the bivalves, by doing the growth increments. This is slightly more complex, and it requires human input, which is, I get ribbed by my colleagues quite a lot because I like data processing. And the reason I like data processing is because I like eyeballing every single specimen in my data set and making sure that it is what I think it is, because I hate plotting something and having to sit there and go squinting at the outliers and go, what is that? What taxon is that? Is it really there? Or did I do something wrong? So I like this kind of manual human in the loop processing. So what we do is we read this all in, but because the bivalves were all scanned at random orientations as part of a different project and at different sizes and shapes, they're not aligned to each other. Alignment is very key for this. So in order to do that, we have to manually align these now. And the way we do it, I haven't demonstrated it here because it takes a lot of time, but effectively we plot different projections of the data, and you look for the projection that gives you the largest X, Y, and the flattest Z versus R arc length, and where yaw and pitch are plotting independently. And then you consider that to be the correct rotation, and you build your own little data file, which you can then read back in this line and apply it. So now we have, these are the alignments of 9 little bivalves. As you can see, they're all digitized in different spaces, but we bring that in together. 173 is a spondylus, in case anybody likes bivalves. What else have we got here? Oh, 188 is Lyrophora latilarata. If you like venerids, that's probably one of the prettiest. And then we read in what I just showed you with the Nipponites. There's a command line, there's a little piece of terminal code that will do the skeletonization, and it outputs an OBJ file, which you can either plot, as I showed you in MeshLab, or you can extract the vertices from that and combine them into a CSV and then pull them all in together. And now this is the thing. This is extracting the yaw, pitch, and roll. You end up with quite a large data set that gives you yaw, pitch, and roll. It also gives you, for any given arc length, it gives you X, Y, Z. It gives you the tangent, the arc length, how big the tangent is, and then it gives you normalization and the specimen name itself. All you're really interested in most of the time is yaw, pitch, roll, and ID. So you can start plotting these. You can see that the polar coordinates are, the specimens that were done with polar coordinates are beautifully smooth because, of course, they were interpolated by a spline. These interesting wiggles are bivalve. Also, most of these are the same taxon, which is why they look so similar. They are Struthularia, looking at those numbers. So they're a Struthularia. They're a little whelp. The RDS things, these downwards wiggles, these are all bivalves, which is really interesting. And then these high resolution ones are the ones that we did with the mean curvature skeletonization, and something fascinating is happening here. I have 1, 2, 3 Sirion, Cetarium, which was actually like the Cetarium that had derailed coiling, and then Niponites. So there's something going on here, and pitch is all over the place, particularly when you get to these high resolution things, which is fascinating. So already you can see variability in pitch and yaw and how they differ in the same specimen, and why they're not why, but you can see that they're doing different things. And so if I now go back again, sorry, I'm flip-flopping an awful lot here, but I'm nearly done, I promise. The next steps that we're planning on doing are a comparison of these different data capture methods to make sure that we do start getting the same answers from the same things, which is very important, right? You don't want your data capture method to be driving your answers. We definitely want to downsample the skeletonizations because those are very noisy. We need to compile a larger data set of mollusks, which is in the works, and then write the paper, hopefully this year, and then get it out next year because it is very, very close. So in summary, I've got really 2 points, and they're kind of related, and one of them isn't even really new. So starting with the second one, which is comparing shell shape in terms of the spiral means you can compare very disparate shells while respecting homology. That point is not new to me. That is Dave Raupp's entire point in that 66 paper, and I feel like we have spent a very long time proving that that was true and that it's useful. But I think the wider point that really sums up what I was trying to get at by giving this talk is that what's desirable is that you have a rigorous analysis that's biologically interpretable. And yes, and GM is a very good way of getting that. But if GM is not giving you the results that, like, not giving you results with definition or usefulness, it might be that GM is not capturing what you want it to capture, and you need to look for a different method of capturing shape. But you still want it to be as rigorous and as biologically meaningful as GM can be. It's very aspirational. So some useful things. There's code here to run the fixed reference frame analysis. So that will take you to the data dryad repository for that paper. I went looking for that paper the other day, and it's paywalled, weirdly, which I didn't think it used to be. But anyway, the data dryad repository is not, so you can get that there. My email address is here. If you want the paper and can't get behind the paywall, I'll very happily send it to you. And if you have heteromorph ammonites or weird snails or anything else squiggly, and you would like to study how it squiggles and you want to talk to me about that, I love talking to people about wiggles, please email me. There's also just a little bit of extra fun, a slightly different take on how do you measure things that are the same but different. This one for bivalves, and it does use geometric morphometrics, but it's about how to align bivalves, which are generally very, let us say, specific homology depauperate in their shells, even though their shells are undoubtedly homologous. Some references, and this is Roma, who could not be here today because he's in Australia and hopefully asleep right now, bless him. But without his mathematical know-how, none of this would have happened. If anything works, it's because he made it work, and if anything falls over and fails, I probably got it wrong. And with that, I will ask if anybody has any questions.